So good afternoon, everyone. My name is Jeff DeLong. I'm a support scientist from the USDA Agricultural Research Services. And I want to thank you guys for the opportunity to speak today. Today, I want to speak with you about some of the research I'm involved with. It's a project that's funded by both the Washington State Commission for Pesticide Registration, as well as the Washington State Red Raspberry Commission. So my research collaborators on this project include Dr. Shakardar, who's located at Washington State University Northwestern Research Extension Center up in Mount Vernon. That's where my lab is located as well. And Dr. Virginia Stockwell from the USDA-ARS Horticultural Crops Research Unit in Corvallis, Oregon, that's where my program is actually directed from. So this project aims to characterize botrytis populations on red raspberry in northwestern Washington by evaluating fungicide-tolerant phenotypes and investigate how these attributes are changing in respect to time. So this is a 3-year project, and we're kind of bringing year 3 to a conclusion. So without further, let's go ahead and talk. Botrytis cinerea. Botrytis cinerea is a fungus. It's mold. It is causal agent of gray mold and fruit rot. It's a necrotrophic pathogen, meaning that it actually has to kill its host tissue in order to digest it and consume it. It has a global distribution. It is absolutely ubiquitous. It's on all 7 continents. It affects over 200 economically important crop hosts. It causes both pre- and post-harvest diseases, and management relies heavily on fungicides, but resistance has also been reported quite a bit in it. And then it's also probably worth mentioning that a lot of the conventional red raspberry growers in northwestern Washington routinely experience anywhere between 5 to 30% annual yield loss and high disease pressure years due to this fungus. So the objective of this project were rather straightforward. We wanted to look at developing rapid assays to detect fungicide tolerance. We also wanted to look at the tolerance and botrytis to some of the newer FRAC 7 fungicide chemistries. We also wanted to kind of evaluate genetic methods to trace origins of botrytis in red raspberry fields in northwest Washington. In other words, what we wanted to know was, is botrytis established in the overwintering in the fields in the form of cane botrytis infections, or does it come in from other fields and other plants? And so just to cover a little bit of terminology here as we also go along, FRAC stands for Fungicide Resistance Action Committee, Action Class. And each number that's typically associated with it specifies the fungicide binding target. So for instance, in FRAC 7 here, whether it's cell membrane synthesis, FRAC 7 targets cellular respiration in the fungus. This is how it actually affects the pathogen. So our sampling locations took place in Whatcom County. We collected from the year 2022, 2023. We collected from 12 commercial red raspberry fields, as indicated by the little red dots here. My mouse is up here somewhere. These little red dots up here, most of them were in Linden County, or in Linden. We collected both asymptomatic and non-symptomatic disease tissue. And we collected 3 stages were sampled from each year. So we collected a stage called overwintering that typically took place in mid-January, at bloom, mid-May, and of course around harvest in mid-July. So our sampling criteria, again, straightforward. We collected approximately 30 to 40 samples were collected from each site in a randomized in-row collection pattern across the field. So as you can see in the image here on the right, we would go down the rows, we'd collect some from on the left, we go down 15, 20 plants or so, collect some on the right, and we complete the row, skip a couple rows, come back the opposite direction, and we'd zigzag our way through the entire field. Now, collecting in this manner allows us to get a good representation of the disease population in the field, as well as the disease pressure within the entire field. So some of the cultivars, some of the popular cultivars that we collected for part of this project was Meeker, Wakefield, Wake Haven, and Cascade Premier. And we also collected approximately 1,200 botrytis isolates. So, having done all those collections, what exactly does that mean? Well, it's worth mentioning that the type of tissue we collected, whether it was asymptomatic or non-symptomatic, was correlated to the collection stage or time point. So when we went out in winter, there's not a lot of foliage or anything like that that's happening. So most of our collections consisted of cane botrytis. So as you can see here on the image on the left, you have canes of raspberry tied up. And I don't know if you can see it in the image, but there's little black dots that's going up on the cane. These are called sclerotia. This is the overwintering structure of the fungus. These are chemically resistant, UV resistant, temperature resistant. This is what the botrytis kind of tightens down in the winter with. So we would collect those in the wintertime. And at bloom, of course, we'd be collecting flowers, infected petioles, infected leaves, those sort of things. And then, of course, around harvest, we're collecting typically fruit. So once we collected all of these, the tissue, we would bring it back to our lab. We would incubate any sort of asymptomatic tissue, and then we would, using aseptic technique, we would transfer it over to cultured media. So on PDA plates, as you can see here on this particular, we have Botrytis growing on a potato dextrose agar. We then did a little bit of monocondidial culturing. So essentially, we made a spore suspension, transferred that over and onto water agar, and we're able to actually isolate or do monocondidial cultures of the particular fungus. You can see here in the center picture, this is actually a close-up of sporulating. So as the fungus actually grows across the plates, the hyphae grows, it creates these structures called conidiaphores. It's a fruiting body. And on the fruiting body, of course, you have the conidia or the spores that grow. If you can kind of imagine the conidiaphores as a tree and the conidia as leaves on that tree, that's kind of the structure it forms. And so we're able to actually plate those on water agar. You can see these little black specks here and a little bit of squiggly lines. So those are single spores. We're able to take a needle under a microscope and actually isolate all those and get monocondidial cultures. Now, this is important, this procedure. I know it sounds kind of tedious, but it's important to clean up the fungus because this ensures us that we've isolated pure botrytis and we have monocondidial cultures. It's not a mixed population of multiple spores or anything like that. So we have a nice clean genetic lineage. And it also kind of helps us tease out some of the other fungal pathogens, the secondary pathogens like Alternaria, Cladosporium, some of the zygomycetes, zygomycetes or anything like that we might find in the field. So going back to our objectives, looking at objective one, developing rapid assays to detect fungicide resistance, and of course, objective two, tolerance to newer FRAC7 fungicide chemistries. These two objectives kind of correlate in one, and I'll kind of show you how we kind of approach dealing with these. So this slide may give some of you kind of PTSD from high school biology. It's not a big deal, but I'll kind of walk you through it. But essentially what I want to show you here is how FRAC7 fungicides are targeting the fungus's ability for cellular respiration. So on the left there, you can see we have a fungal cell. So it's just a normal cell. And because they're eukaryotic, inside that cell, we have mitochondria. If you guys remember, mitochondria are the powerhouse of cells. You know, it's what creates, what gives us energy, what gives the funguses energy. And so on the inner membrane of mitochondria, there's what's called a mitochondrial respiratory chain. And I won't get too deep into the details of it, but there's a binding site, a molecule called succinate dehydrogenase complex 2. And this complex is made up of 4 subunits, subunit A, B, C, and D. Now, the way FRAC7 fungicides work is they're succinate dehydrogenase inhibitors. They actually attack or they affect the succinate dehydrogenase complex 2, whether they're dealing with the subunit A, B, C, or D, and thus they make the complex to make the succinate dehydrogenase ineffective, and thus the mitochondria can't function properly, and thus the fungus can't function properly. So, with that, kind of show you guys how we actually dealt with seeing how these FRAC7s are actually affecting our funguses. So we determined that the quickest and most rapid way for dealing with this was doing a spore germination assay. So we took our monocondidial cultures, Botrytis spores, and we placed them on solidified media with and without fungicides. This was in 12 and 24 well plates, shown on the left-hand side here, 12 well plates at the top, 24 at the bottom. And we examined the spore germination, single spore germination, and growth with a microscope after 16 to 18 hours growing in the dark. So in the bottom table here, you can see we've got our different fungicides, the trade names associated with those fungicides, and then we tested 2 FRAC groups. We tested FRAC group 9 and FRAC group 7. So our fungicides we used were Sopranido, which is typically known as like Switch or Vanguard, Pyrimethanol, typically Scholar, Luna Tranquility, Isofetamide, which is Kinja, Fluxaperoxide, typically seen in Maravin or Xinium, Boscolid, Pristine, and of course Fluopyram, which is seen in your Lunas. So how did this work? How did we evaluate what we saw within the fields once they'd grown up? Well, with every single assay, we had a well with no fungicides in it, and this is called our positive control. So we would take our spore suspension, pour it on there, and then what we would see is, I don't know if you can see the little dots here. Again, these are single spores, and the little squiggly lines, those are called the germ tubes. And that's essentially what the fungus produces to infect its host. And that's what it's trying to produce on this artificial media. It's producing these germ tubes. There's no fungicide, so it's doing what it's supposed to do. And then, of course, we'd use the same spore suspension with our particular fungicide of interest that we were looking at, and we would get one of 3 results. So we would either we would see little to no germination. or growth of the fungus. So you can see the spores here with no germination tube, and it's always getting compared to the positive control. So we would rate these as sensitive, and then we would also see some that were actually growing moderately, so just a little bit of growth, but slightly reduced. So quantitatively and qualitatively, the germ tubes aren't growing in comparison to the positive control. We'd be able to rate those as moderate. And then, of course, we had great germination, very similar to the positive control. So as you can see here, qualitatively and quantitatively for the germ tubes that's growing out of these spores, it looks very similar to the positive control. And we could rate that. We'd feel successful at rating that as tolerant. So with all that data, I want to go over some of the fungicide tolerance we're actually seeing. So one of our main data set, I know we collected approximately 1,200 isolates. We were able to actually function and work with 834 of them. Now, the sample set was replicated twice. So this was gone through, we did these twice. This consists of all 12 of our fields, and we looked at 6 time points. So again, overwintering, our three stages that we looked at: overwintering, bloom, and harvest. This was done for 2022 and 2023. So let me take you through this chart just real simply. On the y-axis, we have frequency of fungicide tolerance. And across the x-axis, we have our different fungicides with the different discriminatory doses underneath each of those. And then in the green lines, the green represents our 2022, and the raspberry color lines represent our 2023. And they also transcend or descend or ascend, from overwintering through bloom into harvest for both of these in this thing. And so the takeaway message from this particular data set is that we're seeing a reservoir resistance that's overwintering. In other words, let's say, for example, we look at fluopyrim here. So in the overwintering for 2022, we have, what looks like approximately 65% tolerance to fluopyrim. When we move up into bloom, we're seeing an increase in that. So the trend is increasing. And then at harvest, of course, it's still continuing to increase. But when I say reservoir resistance, so this ends in July, we start testing and screening again this exact same field for isolates. In 2023, we're starting out where we ended the year 23. So that resistance isn't decreasing. The trend continues on. It stays stable, and it's also increasing. Another trend we see in this particular data set is a bump in the number of isolates that are resistant to a number of chemicals. Example, flexipyroxid. Flexipyroxid isn't regularly used in raspberry fields, not typically. So the fact that we're seeing 25 to 30% resistance in flexipyroxid may suggest that we might be seeing a little cross resistance. We kind of looked at that and addressed that in some of the little bit more data. Moving in the future, I'll show you in just a second, but that's definitely a trend that we noticed. And then, of course, last, kind of just the outlier, the standing out here is fluopyrim. I mean, we've got almost 85% tolerance in fluopyrim. This chemical was introduced semi-recently, and we're seeing high levels of resistance. We're wondering if it's tied to pyromethanol, which is, both of those particular Frac 7, Frac 9s are found in elute tranquility or something like that. So I'll kind of walk you through this chart here, but due to the variability and reduced sensitivity to many of the isolates we saw in screen, in particular the Frac 7s, we wanted to investigate the frequency of tolerance to multiple chemicals within a single frac group, specifically Frac 7. Right, which means that's exactly what we're seeing in this graph here. So let me walk you through this. So on the y-axis, we have our frequency of fungicide tolerance. Across the x-axis, that's moving here, we have our different treatments. So we have sensitive. We have your 4 Frac 7s that I had showed you before, just in the previous slide: isofetamide, flexipyroxid, boscalid, and fluopyrim. And then we started looking at multiple combinations. So when we have the same data set after we had rated everything, we look at each of the isolates and, we line everything up and we're like, okay, well, it's not just resistance to boscalid. It's not a single resistance. There might be isolates that are showing resistance to boscalid and fluopyrim only, or flexipyroxid and fluopyrim, or 3 particular Frac 7s, as you see here with isofetamide, flexipyroxid, and fluopyrim. And even, when we look at all 4 of the Frac 7s we use, isofetamide, flexipyroxid, boscalid, and fluopyrim, we see an increase here. And then, of course, moving across the X plane from your point of origin, starting at overwintering 2022 to the actual point of origin on the graph, moving forward, we move through all of these time points. So that's showing with the line. And so what this data is showing is it's the same trend of fungicide tolerance to some of the same single chemistries. So while you may see 80% in fluopyram when you're looking at it single resistance. When you compare it and you're only saying, oh, but you're only seeing 50% resistance here. There's a reduction in that. It's not a reduction. The resistance that we were seeing in fluopyram, we're now counting into that fluopyram as getting grouped in with a multiple fungicide tolerance. So when we look at the multiple fungicide tolerance to 3 or 4 of the Frac 7s, we're seeing trends that are increasing up to 20%. So you can see here, like in this particular bar here, for all of the time points we tested for this particular 4 different fungicides, we're seeing an increase up to 20%. We also see that with isofetamide, fluxyperoxide, and fluopyram for around harvest of 2022 and the overwintering of 2023, where we screened, we're seeing 20% increase. So when we reevaluate when we reevaluate our data to encompass all the Frac 7, it's not decreasing in a single chemistry, but this accounts for all of the multiple fungicide tolerance we're seeing. So now, having observed this trend to multiple fungicide tolerance in respect to Frac 7s, we're curious to know, why is this happening? How is this happening? What's going on? So bringing you guys back to this particular figure, and this is the reason I showed you, in order to address this question, we sequenced some of the isolates with some of the distinct multiple fungicide tolerance phenotypes. And what we saw was we took a subset 28. Now, these are randomly selected based off the fungicide tolerance profiles. Again, we had some that were completely sensitive to all 4 of the Frac 7s, and then we had some that were all over the place. And once we sequenced them, we got a mutation P225F at 43% of our population, mutation N230I 25% of our population, mutations H272R at 11%, mutation H272V at 4%, and of course no mutations accompanying for 18%. Now this was, again, this is no surprise here because we did include isolates that were sensitive to everything. And one of the things to take away here is that, the H227, whether it's R, V, or Y mutation, this is commonly associated with just boscalid tolerance only. And isolates possessing mutations P225 and N230I are extremely often, exhibit shared tolerance to more than one SDHI fungicide, typically 3 to 4, and if not even more, that we didn't even test in this particular thing. So previously, a lot of the Frac 7, 9, and 17 resistance was documented by Tove and Peever's lab in Northwestern Washington. And it's worth noting that majority of what they screened, they had observed H272Y mutations had comprised 62% of their population, and H272R was comprising of 38%. We don't even see the H272Y population, and we're only seeing a bit of H272R in 11%. So, why are they only tested boscalid in one of their Frac 7s? We continue to test boscalid, but we're also testing a lot of the other Frac 7s, and we're seeing multiple fungicide tolerance among these Frac 7 chemistries. So, with all that said, I'll leave you with one last slide, and this goes out to my growers. So, one might say, well, I'm not seeing these resistant failures you're talking about. I'm not seeing, maybe I don't have resistance in my field. Maybe I don't even have a botrytis problem in my field. So as another project that this data led us to, we got a grant with the Northwest Center for Small Fruits Research, and it basically functions to look at, is overwintering botrytis inoculum in the field, does it serve as the primary inoculum source for the next upcoming season? And part of that, so we're doing a lot of collections for that, multiple different tissue types, and we have done monoconadial isolations for approximately 1,000 of them. But out of flower and ripe berry, we collect asymptomatic fruits for those. So there's no disease on that. So you got approximately 400 of each, almost 800. Of the asymptomatic tissue that we got from flower and ripe berries, we're seeing 67% recovery of botrytis from flowers or something that doesn't even have botrytis, that's asymptomatic. We're recovering botrytis 47% of berries with no known botrytis symptoms or even signs on it. So what that tends to suggest that the pathogen is in the field, and the resistance potential for that pathogen is there. It is growing, so it is a concern for us. So takeaways, we are observing tolerance developing to some of the newer Frac 7 and even Frac 9 fungicide chemistries. The trend to increase tolerance to multiple Frac 7 chemistries can be noted in the different site mutations we're seeing in the populations of those. We're seeing resistance in botrytis collected in the winter months. In other words, tolerance is persisting into the next growing season. It's not stopping. We will continue to monitor fungicide tolerance, And we are also going to continue to look at some of the genetic analysis of the winter Botrytis populations compared to those from bloom and fruits and determine relatedness. So with that, I simply want to acknowledge and thank some of our collaborators on this project. Again, Washington Red Raspberry Commission, Washington State Commission for Pesticide Registration, and Washington State University collaborators. Thank you very much. And the biggest thank you of all, thanks for all our collaborating growers who are putting your fields on the line and trusting in us to kind of help you guys. So we really appreciate you. And with that, I thank you guys and I'll take any questions.
Questions?
Jeff, I was curious about that really high incidence of finding botrytis in asymptomatic tissue. And I was thinking about what you'd said earlier in the talk that it's botrytis is a necrotrophic organism. So if there's nothing on that asymptomatic tissue that's dead, how does the botrytis survive there?
So we're kind of considering if botrytis is, let's say, infecting flower and it's going into the ovum of the flower or something like that, and it's actually in the green fruit. We know botrytis is actually it's been shown that botrytis lives in the green fruit of blueberries. We're suspecting it may actually still be living in the green fruit of raspberries or something like that. It just may be a latent infection. It's just not popping up at that point. If we incubate it, once we incubate it, it comes right out.